Thank you to Squishmallow for sponsoring this week's episode.
Hi guys, welcome to Catherine's Anatomy and Physiology podcast. Today we're going to be talking about three subsystems: neurology, respiration, and phonation. We'll be going over some anatomy and physiology of these three subsystems and talking about how they relate to patients with Parkinson's disease. Alright, so we're going to start off with neurology, and we're going to go over the anatomy of the brain and the lobes of the brain. So today I have my Squishmallow assistant here to help us go over that. So starting off, just to orient us, this is the front of the brain, and posteriorly we have the back of the brain. The brain is split into two hemispheres, a right hemisphere and a left hemisphere. So now let's dive into the lobes. So starting off with the frontal lobe, which as you can assume is in the front, it's located right here. Some landmarks of the frontal lobe are the precentral gyrus, the motor strip, which is located here in green, the prefrontal cortex, and Broca's area. Broca's area is particularly important for speech and language, and it's located on the left frontal lobe right here. Next up, we have the temporal lobe, which is located on either side, right above where the ear would be. And the landmarks of this lobe are Hessler's gyrus, the primary auditory cortex, and Wernicke's area. Wernicke's area is another area that is particularly important for speech and language. It's located on the left hemisphere of the temporal lobe, right here. Next up, we have the parietal lobe. You can go posteriorly to check it out. The landmarks of this lobe are the postcentral gyrus, the sensory strip, which we can see here in blue, the supramarginal gyrus, and the angular gyrus. And then lastly, for lobes of the brain, we have the occipital lobe, and the landmarks of this lobe are the calcarine sulcus and the primary visual cortex. So now that we've gone over the lobes, I want to draw our attention to two major communication pathways within the brain that are particularly important. So the first one is the corpus callosum, which allows for communication between the right hemisphere and the left hemisphere. It runs right down the brain medially, and it allows these two hemispheres to communicate. The next one is the arcuate fascicula, which allows for communication between Broca's and Wernicke's area. If you remember, as I previously mentioned, they're both in the left hemisphere of the brain. So we can see that Broca's area right here is able to communicate with Wernicke's area right here because of the arcuate fascicula. Okay, so right now we are looking at the right hemisphere of the brain. This is a sagittal cut of the brain, so we're going to take a look at some of the structures within. So starting off with the cerebellum in green, this is also known as the mini brain. Next up, we have the brainstem here in orange, which can be separated into the midbrain, the pons, and the medulla oblongata. Next up, we have the hippocampus in this bright pink right here. Then in front of the hippocampus, we have the amygdala. In yellow, this little structure right here is the hypothalamus. In sparkly blue right here, we have the pituitary gland. In red, we have the thalamus, and in blue, we have the basal ganglia. And I want to really orient our attention to the basal ganglia because this will be particularly important when talking about Parkinson's disease. Next up, as we work through the anatomy of the brain, we're going to talk about the cerebrovascular system, which is really just a fancy word for how the blood gets supplied to the brain. All right, so I have this visual that I think is really helpful in understanding how this blood supply runs from the heart, along the spinal cord, and into the brain. So this is an inferior view, and then over here we have just a view of the circle of Willis. So I highlighted some terms I want to run through that are important. So first we have the anterior cerebral artery, which is this one right up here at the top. And that is what you will see right here and the most anteriorly in the brain. Next up, we have the anterior communication artery, which is shaped like a little U right there. And again, you can see it just posteriorly to the anterior cerebral artery right there in the brain. Next up, we have the middle cerebral artery, which are these kind of thicker looking arteries you see right here. And again, you can see them coming in laterally and running medially right there. And then one that's not listed over here that I still want to draw our attention to is the internal carotid artery, which is the ends of the middle cerebral artery right here. As you can see, I highlighted it. And then again, if you look over at the brain, you'll be able to see where it would be. Next up, we have the posterior communication artery. This is running right here, and you can see it coming down posteriorly to all these other structures we've been looking at. Next up, we have the basilar artery, which looks like a big centipede right here, and you can see it very noticeably right here on the brain when you're looking at it from the inferior view. All right, so now that we've addressed some of the larger structures within the brain, we're going to take a look at the nitty-gritty of the brain, which is the neuron. We're going to do a little bit of neuron anatomy, and then we're going to talk about how messages are sent through the neuron with action potential. So here we have a diagram of the neuron, and starting out here where the message is received at the dendrites. Then you can move into the soma, also known as the cell body, which contains the nucleus within it. If you look down here, you can see in red there is an axon connecting the soma to the axon terminal. The axon is covered in these little bubble-looking structures known as myelin sheath. And at the end of the axon over here, we have the axon terminal in pink. And at the end of the axon terminal, we have the terminal buttons. And then the space between the neurons, so where this neuron terminal button would be meeting the dendrites of another neuron, is called the synaptic cleft, which you can see a close-up shot of here. So the process of neurons exchanging information and transporting signals is called action potential. According to Differding's lecture on neurology, we can understand action potential as the cell membrane being stimulated. There's a change in the electrical potential, and it permits ion exchange. So basically what this means, and I'm going to use our diagram that we just went over to help orient us. So the message will be received from the postsynaptic neuron via the dendrites, and it will travel towards the axon, where it will move quickly because it is insulated in that myelin. So when the neuron is stimulated by the signal, it becomes depolarized, and as it's depolarized, the signal will travel down the axon through the neuron, and eventually it will start to repolarize back to its resting state. There will be a brief period where it overshoots, becomes too negative, and reaches hyperpolarization, but it will restabilize, and when the signal reaches the axon terminal, it will be released by these terminal buttons. That's where neurotransmitters come from, right down here. Now that we've gone over the anatomy and physiology of neurology, I want to share some research I did about how neurology can be impacted by Parkinson's disease. So according to Penn Medicine, the principal features of Parkinson's disease can be attributed to the loss of dopamine. And this dopamine is produced by neurons projecting from the substantia nigra to the motor source, which, if you remember, we have our motor strip right here. So knowing that Parkinson's disease can cause a decrease in dopamine, Stossel, Lierse, and Strafella wanted to conduct research to see how the loss of dopamine could affect the basal ganglia. If you remember, the basal ganglia was the structure I pointed out in the subcortical structures earlier in the video. So in their study, they wanted to use PET imaging to see how the basal ganglia was affected. And through their neuroimaging, they were able to provide strong evidence that the damage done to the basal ganglia could be considered a major determinant in the pathophysiology of primary dystonia. And dystonia is a common symptom for people who have Parkinson's. I'm going to go more into detail about spasmodic dystonia in the phonation unit. However, what you do need to know about dystonia now is that it occurs from failure of the basal ganglia and the filtering that enables voluntary movements. So basically what is happening with neurology and Parkinson's is people with Parkinson's also suffer from a severe decrease in dopamine in their brain and the neurotransmitters, and that can cause adverse effects to the basal ganglia, which can then cause adverse effects to their fine motor control. All right, so the next unit we're going to talk about is respiration. And from Differding's respiration lecture, respiration can be defined as the exchange of gas between an organism and its environment. So I have this image here to help orient us to some of the basic anatomy before we dive into the details. So starting up here, we have the nasal and oral cavity, which is where oxygen is going to enter the organism. It's going to travel down the windpipe into the trachea, and then it is going to enter the bronchi, which are these two big larger offshoots off of the trachea. From there, it's going to filter into the bronchioles, which are these little offshoots off of the larger ones, and then end up in the alveoli, which are these little dots at the end. And the walls of the alveoli are where the gas exchange is going to occur, so O2 into CO2, and then it is going to exit through the same tract it entered, so out through the bronchi, through the trachea, and then exit through the oral or nasal cavity, where it will be released back into the environment. So when talking about respiration for speech, we can start by thinking about the diaphragm. The diaphragm is located right here. It's the primary muscle for respiration, and it contracts down and forward for inhalation when we are about to speak, so as to increase the size of the thoracic cavity, which is this. So while the diaphragm is contracting down and forward, the accessory muscles of inhalation are contracting anteriorly and posteriorly, so front and back, to also help in making the thoracic cavity bigger. And the reason we want the thoracic cavity to be so much bigger on inhalation for speech is because we want to get as much air in as possible so that we can use it to aid in phonation. So after we have inhaled for speech and we begin to exhale, this is where the accessory muscles of exhalation are going to come into play. So the exhalation, So now that we've inhaled, obviously we are going to need to exhale. This is where the muscles of forced expiration are going to come into play. They are going to help release that CO2 back into the atmosphere. They're going to pull the rib cage down and compress the abdomen beyond what it would be doing during normal rest breathing. Specifically, the internal intercostals are going to pull the ribs down. And while this is happening, the diaphragm is also going to relax. It's going to go up and back into its resting position. This is going to overall decrease the size of the thoracic cavity. So, pulled from Differding's lecture, there are a few functions of the pleural lining that are really going to aid in respiration. The pleural lining is able to allow for reduced surface tension of the lungs. It provides lubrication between the lungs and the walls of the thoracic cavity, and it prevents the visceral and the parietal from sticking to each other. And all these functions are going to aid in smooth and effective respiration, which is very important for speech. So the main difference with speech, respiration for speech, is that both the inhalation and exhalation are an active process, whereas for rest breathing, just the inhalation is an active process. Speech respiration also requires the use of the accessory muscles of inhalation and exhalation, and those are because we really need to increase the volume of the thoracic cavity when we are using respiration for speech. To understand respiration, we need to take a look at Boyle's law, pressure, and volume. So from Differding's lecture on respiration, we know that Boyle's law states that volume and pressure have an inverse relationship. So as volume increases, pressure will decrease, and as volume decreases, pressure will increase. Pressure is defined as the force exerted on the walls of a container. So in this case, the container we're talking about is the thoracic cavity, and gas is known to move from an area of high pressure to low pressure. Boyle's law can explain how when the diaphragm contracts and the accessory muscles of inhalation contract, we know that the thoracic cavity starts to get bigger. Because the cavity is getting bigger, the volume is increasing, which we then know in turn will cause the pressure to decrease. Because the pressure is decreasing, the atmospheric pressure will want to leave the atmosphere and come into the thoracic cavity. This is because gas will move from an area of high pressure to low pressure. So now the gas is in the thoracic cavity, and eventually the diaphragm will begin to relax. The accessory muscles of exhalation will begin to relax, and the volume will decrease in the thoracic cavity. As the volume starts to decrease, the pressure in the thoracic cavity is going to start to increase. So therefore, the gas is then going to exit the body back into the atmosphere where the pressure is lower. All right, so as we know, the decrease in dopamine can cause motor difficulties for those with Parkinson's disease. A study done by Axelrod et al. Noted that the expansion of the rib cage and the abdomen involves a synchronized contraction of the diaphragm and those muscles we just talked about. People with Parkinson's are also noted having the prevalence of a restrictive respiratory disease. This will cause their contraction to not be synchronized. This can affect their breathing pattern, and the chest wall volume has been found to vary between 28 and 94%. This means that patients with Parkinson's are going to have a more difficult time expanding their thoracic cavity to an optimal size for effective and smooth breathing. The respiratory difficulties that Parkinson's patients often have can cause symptoms such as sleep apnea to develop due to the deficit in effective breathing, and it can also be as severe as causing mortality. So this article notes that the respiratory difficulties people with Parkinson's disease have can sometimes cause a secondary disease to develop, such as sleep apnea, and it can even increase the risk of mortality. The poor ventilation that they have will overall most likely decrease their quality of life while they're suffering from this disease. All right, so now that we've wrapped up respiration, we're going to talk about phonation. And from Differding's lecture, we know that phonation is the voicing that occurs as a result from the vibrating vocal folds. We also know from this lecture that the larynx is the primary structure for this. I have a picture right here for us to look at. And the larynx is located right here above the trachea and below the hyoid bone. So we can consider respiration and phonation a sequential process because we know that we need respiration as the source of power to allow for phonation. We need respiration to be able to phonate, and next up I'm going to go into more detail about how the larynx uses the power from respiration to permit phonation. Okay, so we know that the intrinsic muscles are involved in the production of sound. From Differding's lecture, we can identify intrinsic muscles as the muscles that have both their origin and insertion within the larynx, and they make fine adjustments to the vocal folds, specifically opening, closing, tensing, and relaxing. All right, so first up, we're going to talk about the vocal folds closing. This can be referred to as adduction. It happens via the adductors. A helpful way to remember this is that the adductors add the vocal folds back together and close them off. Their function is to adduct the vocal folds in order to terminate phonation and protect the airway during swallowing. The adductors we want to keep in mind are the lateral cricoarytenoid, the transverse arytenoid, and the oblique arytenoid. The opening of the vocal folds happens because of abduction. This creates space between the vocal folds, and according to Differding's lecture, abductors open the vocal folds for breathing and the initiation of phonation, which means when we start our speech. And the important abductor to know is the posterior cricoarytenoid. Lastly, we're going to go over relaxers. They are responsible for relaxing the vocal folds. They decrease tension and to allow the vocal folds to have pitch variation. And the relaxers you want to keep in mind are the thyromuscularis and the thyrovocalis. All right, so now we are going to talk about the cartilages that are involved in phonation. There are some that are paired and there are some that are unpaired. We're going to use this picture to help us go over them. So starting off with our first unpaired cartilage, we have the cricoid cartilage. It is the base of the arytenoids, so it articulates with the arytenoids right here. The arytenoids are next up. It is a paired cartilage, so there's one here there's one here, and they are the posterior point of attachment for the vocal folds. The landmarks to note are the muscular process and vocal process, and these rock and glide in order to allow us to adduct and abduct our vocal folds. Next up, we have the craniculate cartilage, which is right on the tip of the arytenoids. It's another paired cartilage, and it also aids in the fine adjustment of the vocal folds. Next up, we have the cuneiform, which you can't see too well in this picture, but it would be up here. It is a paired cartilage, and it aids in the stiffening of the vocal folds and the abduction. Next up, we have the thyroid. We're going to look over here to look at the thyroid. So the thyroid is the largest unpaired cartilage, and it rocks forwards and backwards as we phonate. The last unpaired cartilage we have is the epiglottis. It's right here. It doesn't have a speech function, but it does allow us to have a safe swallow. So now that we've gone over some of the anatomy of the muscles and the cartilages, we can consider how they articulate together to allow us to abduct, adduct, as well as tense and relax those vocal folds. Now that we've addressed that, we're going to go over the stages of phonation. So the 3 stages of phonation to keep in mind are initiation, sustained phonation, also sometimes called continuation, and termination. Okay, so next up we have sustained phonation. It's also called continuation, as I mentioned, and from Differding's phonation lecture, we know that this is when the muscles of adduction, which are those muscles that bring them together, allow for the vocal folds to stay in the airstream while the vocal folds vibrate. They have a specific frequency depending on the length, mass, and tension. And we also know that the vocal folds are going to open and close from bottom to top. So if these are our vocal folds, when there is a simultaneous phonation occurring, they are opening and closing both at the bottom and then to the top. And this process is repeating hundreds of times per second. The final phase is termination, and this one is boom. The final stage is termination, and this is when the muscles of abduction, so those muscles that are kind of taking the vocal folds apart, allow for the vocal folds to be removed from the airstream. There are 2 theories that can really help us understand how all these structures are interacting. There are 2 theories that can really help us understand how all these structures are interacting for phonation, and they are the myoelastin theory and Bernoulli's effect. I'm going to go over a brief definition of these and then how they interact to help us understand the physiology of phonation. The myoelastin aerodynamic theory is explained in Differding's phonation lecture, and it can explain the vibration of vocal folds based on their interaction with the air pressure and the vocal fold composition. So we know that the composition of everyone's vocal folds is going to be a little bit different. It's based on the mass and length of them, and the subglottic pressure that builds underneath of the vocal folds in the subglottic space is going to peel those vocal folds open, again from bottom to top. And as they begin to peel apart, the rate at which they vibrate from this pressure is going to be based on the size, length, and mass. Differding's phonation lecture also explains Bernoulli's effect as having a constant flow of air at the point of constriction, and it will cause a decrease in pressure and an increase in velocity. So pressure and velocity will have an inverse relationship at the point of constriction, aka where the vocal folds are getting skinnier or closer together. So what this means for speech is when our vocal folds are abducted or getting further apart, there will be high pressure, but the velocity will be low. This will result in lower, slower pitch voice, and when the vocal folds adduct together, they get closer. This will decrease the pressure, increase the velocity, and there will be a higher pitch and frequency of someone's voice. So these 2 theories can work together to explain how the composition of someone's vocal folds, the inverse relationship of pressure and velocity, will work together to create someone's frequency and can contribute to their optimal pitch, which is the optimal pitch at which they will speak at regularly based on these factors. So now we're going to talk about how patients with Parkinson's can experience phonological difficulties. So an article by Yang et al. Found that more than 90% of patients with Parkinson's disease suffer from hypokinetic dysarthria. And this can present as speech flaws that are typically characterized by increased acoustic noise, reduced intensity of voice, harsh or breathy vocal quality, increased voice nasality, so sounds coming through the nose, mono pitch, mono loudness, speech rate disturbance, amongst other things such as accelerated speech or a decrease in speech syllables. So these speech impairments are caused by impairments to the speech mechanisms, similar to the stiffening of the muscles in the thoracic cavity. The laryngeal muscles tissue can also stiffen, and the vocal folds can begin to harden. This can affect the closure of the vocal folds and increase muscle tone. And I touched on this briefly during the respiration section, but I'm just going to circle back to reiterate that the decrease in respiratory capacity that patients with Parkinson's disease have will also make it harder to phonate, because in order to have sustained phonation, you need to be able to have a steady, sustained breath of air, and if they can't bring in as much air, they're not going to be able to phonate as easily. The Yang et al. Article found that patients with Parkinson's disease presented with a much lower fundamental frequency. This really just goes to show how the tightening of the laryngeal muscles and the decrease in respiratory capacity are contributing to decreased phonological abilities, which makes it more challenging for people with Parkinson's disease to speak, and it might cause their speech to also present with some disorders. All right, so those are the three subsystems I'm going to talk about and how they relate to Parkinson's disease. So just to summarize and reiterate the main points, there is neurology, which is the brain, and the decrease in dopamine can cause motor difficulties. There's respiration, and the decrease in motor capabilities can decrease the muscle ability of the thoracic cavity, which will make it more challenging for them to breathe, as well as changes in their posture. And then this will all contribute to the affected phonatory abilities because of muscle fatigue in the larynx and just overall motor and muscle difficulties throughout the body in the cerebration and respiration. Thanks for watching. I will be back with a second episode next time.